Hello students, let us do quick revision of 30 important figures given in your NCERT book from term to syllabus for the session 2021-22. This is figure 8.1. It is showing stages in the life cycle of Plasmodium. Plasmodium causes malaria. Female Anopheles mosquito is a vector of the disease. Sporozoite is the infected stage of the disease. Three species of Plasmodium are Plasmodium vivax, Plasmodium falciparum and Plasmodium malaria. Out of these three, Plasmodium falciparum causes most serious types of malaria. Parasite enters liver cell, then into RBC where it multiplies asexually. When RBC bursts, a toxic called hemozoin is released and this causes fever. Gametes are formed in the RBCs, female gamete and the male gamete. Further, fertilization of gametes takes place inside the gut of the mosquito. Meiosis occurs and sporozoites are formed, and these sporozoites are stored in the salivary glands of mosquito. Figure 8.2. It is showing inflammation in one of the lower limb. You can see here due to elephantiasis. Elephantiasis is caused by filaria worm Vaucheria. The two species of Vaucheria are Vaucheria bancrofti and Vaucheria malai. The vector of the disease is female mosquito. Figure 8.3 showing ringworm affected area of the skin. You can see here this is the scaly or the patchy skin caused by fungi belonging to genera Mycosporum, Trichophyton and Epidermophyton. Figure 8.4, it is showing the structure of an antibody molecule. Antibody molecule is represented as H2L2. H2 means it has two heavy peptide chain, number 1 and number 2, and L2 means it has two light peptide chains, number 1 light peptide chain and number 2 light peptide chain. Some antibodies produced in our body are IgA, IgM, IgE and IgG. The response produced by these antibodies is humoral immune response as these antibodies are present in the blood. IgA type of antibody is present in mother's milk colostrum produced during initial stage, while IgE antibody is produced during allergic reaction. Figure 8.6, it is showing replication of retrovirus like HIV, human immunodeficiency virus causing AIDS. They have RNA as a genetic material. HIV enters macrophage cells where reverse transcription takes place, means viral RNA will form viral DNA in the presence of enzyme reverse transcriptase. Now, viral DNA incorporates to macrophage cell DNA. Next, viral RNA starts forming. This is viral RNA. As a result, macrophages act as factory of making HIV virus. HIV also enters helper T lymphocytes and produce progeny virus. Figure 8.7, it is showing the chemical structure of morphine which is extracted from latex of poppy plant shown in figure 8.8. Figure 8.9, it is showing the skeletal structure of cannabinoid molecule obtained from inflorescence of cannabis sativa. Figure 8.10 is showing leaves of cannabis sativa. Leaves of cannabis sativa along with flower tops and raisins of cannabis in various combinations are used to produce marijuana, hashish, charas, ganja, etc. Figure 8.11, it is showing flowering branch of datura which has got the hallucinogenic properties. Figure 10.8, it is a typical biogas plant. Indian Agricultural Research Institute IARI and Khadi and Village Industries Commission KVIC developed the technology of producing biogas in India. Figure 11.1, it is showing steps in formation of recombinant DNA by action of restriction endonuclease enzyme EcoR1. For making recombinant DNA, both vector DNA and foreign DNA are cut by EcoR1 enzyme at same site, which is between G and A. This is G and A. This is also G and A of palindrome GAA-TTC, CTT-AAG. As a result, two sticky ends are formed. Number one sticky end from the vector DNA, number two sticky end from the foreign DNA. Now next, these two sticky ends are joined together by enzyme DNA ligase and we get recombinant DNA. Figure 11.2 is diagrammatic representation of recombinant DNA technology. Here vector DNA is plasmid. Both foreign DNA and plasmid are cut with same restriction enzyme at specific point. Then foreign DNA is joined with plasmid by DNA ligase. As a result, recombinant plasmid is formed. This recombinant plasmid is introduced into E. coli cell by the process called as transformation. These bacteria divide rapidly and form copies of DNA fragments in large quantities. Figure 11.3 is typical agarose gel electrophoresis. The fragments of DNA are separated by this technique. DNA fragments are negatively charged. They move towards anode under electric field. Smallest DNA fragments move farther while largest size DNA fragments remain towards the wells. Lane 1. This lane is having undigested DNA, so no movement of DNA fragment is taking place here. While lane 2, 3 and 4, they are having digested sets of DNA fragments. Thus, the fragments formed move towards the anode in lane 2, lane 3 and lane 4. Figure 11.4 is showing the structure of PBr 322. First of all, see the restriction sites present on PBr 322. They are EcoR1, CLA1, HIND3, BAMH1, SAL1, PVU2, PST1 and PVU1. Apart from this, it has got ORI, origin of replication. Then two antibiotic resistant genes, AMPR, ampicillin resistant gene and tetracycline resistant genes. Then it has got ROP, which codes for proteins involved in replication of plasmid. Now figure 11.5. It is showing DNA that separates out can be removed by spooling. Figure 11.6. It is showing polymerase chain reaction or PCR with which copies of gene of interest can be synthesized. Three steps involved in PCR are: number 1, denaturation, it is done by heating. Number 2, annealing, in which the primers are attached. Number 3 is extension, which is done by the enzyme thermostable DNA polymerase obtained from bacteria Thermus aquaticus. As a result, after 30 cycles, billion copies of DNA can be formed. Figure 11.7. 11.7a, it is showing simple stirred tank bioreactor, and 11.7b, it is showing sparged stirred tank bioreactor through which sterile air bubbles are sparged. In these bioreactors, large volumes of cultures can be easily processed to produce large amount of bioproducts. Figure 12.1a and 12.1b. 12.1a is showing cotton ball destroyed by bollworm, while B is showing fully mature cotton ball. To save cotton from damage caused by bollworm, genetically modified crop BT cotton is produced. Figure 12.2a and 12.2b. A is showing roots of typical tobacco control plant, while B is transgenic plant roots 5 days after deliberate infection of nematode, but protected through novel mechanism. This mechanism is RNA interference, where host plant generated double-stranded RNA triggers protection against nematode infestation. Figure 12.3. It is showing maturation of proinsulin into insulin. Proinsulin has three peptide chains: peptide A, peptide B and peptide C. When peptide C is removed, then insulin is formed. Insulin has peptide A, peptide B joined together by sulfide bonds. An American company, Eli Lilly, in 1983 prepared human insulin by using plasmid of E. coli by recombinant DNA technology. Figure 13.1 is showing biome distribution with respect to annual temperature taken on y-axis in degrees Celsius and mean annual precipitation taken on x-axis in centimeter. See the location of biomes as per temperature and annual precipitation. These are deserts, grasslands, tropical forest, temperate forest, coniferous forest and Arctic and alpine tundra. Figure 13.3. It is diagrammatic representation of organismic response to abiotic factors. According to response of organisms to abiotic factors, organisms may be: number 1, conformers. They change internal conditions as per external environment. Second, regulators. They keep their internal conditions constant. Number 3, partial regulators. They have evolved the ability to regulate but only up to limiting range, beyond which they are simply conformers. Figure 13.4. It is representing age pyramids for human population. For constructing age pyramid, number of individuals of 3 age groups are taken: pre-reproductive, reproductive and post-reproductive. If pre-reproductive individuals are more and post-reproductive individuals are least, then population is showing expanding pattern. In second case, the population is stable, where the number of pre-reproductive and reproductive individuals are same. In third, the population is showing declining pattern, where number of individuals of pre-reproductive age are less than reproductive age at a particular time. Figure 13.5 is showing the factors affecting population density N. Immigration I and natality B increase the population density, while emigration E and mortality D decrease the population density. Figure 13.6, it is showing the population growth curve. Graph A, it is J-shaped graph. It is obtained when resources are unlimited in nature and responses are not limiting the growth, and this plot is showing exponential growth. Graph B is S-shaped. It is obtained when resources are limited in nature and responses are limiting the growth. Here the plot is logistic. While K, see the K is the carrying capacity, which is the maximum population size of a species that can be sustained by specific environment. Figure 13.7 is showing mutual relationship between fig tree and wasp. In A, the fig flower is pollinated by wasp, and in B, wasp is laying eggs in fig fruit. Figure 13.8 is showing B, a pollinator on orchid flower. Figure 15.1 is representing global biodiversity. Graph 1 is showing proportionate number of species of invertebrates. These are insects, mollusks, crustaceans, other animal groups. Figure 2nd is showing proportionate number of species of vertebrates, fishes, amphibians, reptiles, birds and mammals. Figure 3 is showing proportionate number of species of plants, fungi, algae, lichens, angiosperms, ferns and allies and mosses. Figure 15.2. This is a graph showing species area relationship. Area is taken on x-axis while species richness is taken on y-axis. The species richness increase with increase in area explored, but only up to a limit, and the graph turns out to be a rectangular parabola. But on log scale, the relationship is linear. You can also note down the equation for both the graphs. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.